Bismillahirrahmanirrahim. In this video, we are going to discuss the provisional matrix. The references for this video are following. Another type of extracellular matrix that contribute to the organization, physical properties and function of the tissue is provisional matrix. This is the temporary extracellular organization of plasma-derived matrix protein and tissue-derived component that are accumulated at the site of injury. For example, hyaluronic acid, tensin, and fibronectin. They associate with pre-existing stromal matrix and serve to stop blood or fluid loss. So, the primary function is to seal the wound. Then they also support the migration of cells, the leukocyte, endothelial cells, and fibroblasts to the site of the wound, so they support migration and they also initiate the repair process. So they are implicated in the tissue repair, the start of the tissue repair. There are various adhesive glycoprotein that are present and they work as a glue for the extracellular matrix and the cell. These consist of fibronectin, tensin, that other name of tensin is cytotectin, and thrombospondin. Fibronectin, that is derived from the word nectar. Nectar means to bind. This fibronectin is the best characterized glycoprotein, adhesive type of the glycoprotein that is present in the extracellular matrix, and it has binding properties for other cells and extracellular matrix. There are two types of this fibronectin: the plasma type of the fibronectin and the tissue type of the fibronectin. As like other proteins in the plasma fibronectin is synthesized by the liver cell and it is trapped in the basement membrane, such as during filtration through the renal glomerulus. Tissue fibronectin is formed by the fibroblast, endothelial cell and other mesenchymal cell and responsible for preventive matrix such as in the fetus and in wound healing. Tensin or cytotectin is the glycoprotein that is associated with fibroblast and appear in the wound after about 48 hours of the injury. It disappears as the scar matures. Thrombospondin is another adhesive glycoprotein that is synthesized by the granules of the platelet. It is an adhesive protein for keratinocyte and platelet, but is inhibitory to the attachment of fibroblast and endothelial cell. The insoluble fibrin is generated through the clotting cascade from the fibrinogen, and the tissue-derived provisional matrix protein are hyaluronic acid and tensin. So, the insoluble fibrin that was generated by the clotting cascade, it is stabilized by factor 13, that is transglutaminase, and this transglutaminase cross-links fibrin to the matrix. Hence, the provisional matrix is internally stabilized and bound to the adjacent matrix by tissue transglutaminase 1 and 2 that is active during the tissue remodeling. In addition to this, there is presence of growth factors, and these growth factors are, one of the important one is the platelet drive growth factor, that are stored in platelet thrombi and released upon activation from there. So the major function of this provisional matrix, as it is composed of adhesive protein, major function is to form a plug, a fibrin clot, to stop the blood and fluid loss. Hence, it is important for tissue homeostasis. Then, it allows leukocyte, fibroblast, and endothelial cells to migrate into the wound. So, it serves as a scaffold for the migration of the cell. There is presence of this platelet drive growth factor and other cytokines that are important for the stimulation of the healing. So this provisional matrix is important in the delivery of the growth factor, and then they are forming a strong connection with surrounding tissue through the factor 13 or the transglutaminase. Then they provide a transitional structure before the granulation tissue forms, so they are important in tissue remodeling. So the function of this provisional matrix are hemostasis, cell migration, growth factor delivery, stabilization of the clot and link to the stromal matrix, and tissue remodeling and support. The quality and persistence of the provisional matrix can influence scar formation. The chronic wound often exhibit persistent disorganized provisional matrix and impaired remodeling. Before closing the discussion on this provisional matrix, I would like to briefly introduce two molecules, fibronectin and hyaluronic acid, that are important component of this provisional matrix. Fibronectin is a matricellular adhesive glycoprotein, and it is a versatile adhesive glycoprotein that is found in the stromal connective tissue and wound provisional matrix. It is a V-shaped homo or hetero dimer that is linked by two C-terminal disulfide bonds. Its interaction are with bacteria, collagen, heparin, fibrin, fibrinogen, and integrin. So its major function are to link matrix component and cell, and to facilitate the cell migration, particularly in thrombi and wounds. Its interaction with the integrin is defined the integrin receptor family through its binding specificity. It forms insoluble cellular form and soluble plasma form. There are two types of this fibronectin, plasma and tissue type. And there are about 24 isoforms that are produced by alternative splicing. A point to remember that both of these, the insoluble and soluble types of the fibronectin, are encoded by a single gene. The plasma fibronectin binds to fibrin and promotes platelet adhesion and stabilizes the thrombi by factor 13 transglutaminases. It promotes keratinocyte migration and attachment in wound healing and aids in repatholization by interacting with the collagen. It is produced by, especially the tissue type, is produced by the fibroblast and assembled in insoluble fibril with integrin and collagen assistance. It is localized in granulation tissue, loose connective tissue, and provisional wound matrix. So, experimental knockout of plasma fibronectin doesn't alter healing. So, there is compensation by the cellular fibronectin. Hyaluronan or hyaluronic acid is the only glycosaminoglycan that covalently not linked to a protein. There is no covalent binding in this. It is a linear polymer of glucuronic acid and an acetyl glucosamine, and there are about 2000 to 25,000 disaccharide units in its chain. They are highly negatively charged due to presence of carboxyl group and extremely hydrophilic. There is no covalent protein link. They are synthesized at the plasma membrane, not in the Golgi like other glycosaminoglycans. They are associated with non-covalently bind proteoglycans such as aggrecan, versican, via link proteins or adapter proteins. They are present in cartilage, joint, vitreous humor of the eye, developing tissue, and provisional matrix. The function of hyaluronic acid is hydration to maintain the tissue turgor and resilience, joint lubrication, and cell migration. They are present in abundance during embryogenesis and tissue morphogenesis, hence they have some role in the development of the tissue. They are key component of provisional matrix and facilitate repair and reepithelialization that emphasizes their role in wound healing. Hyaluronic acid may be increased in inflammation, cancer, and scarring, and these are removed by CD44 monocytes. They are degraded by action of enzyme hyaluronidase, and if there is reduced activity of this enzyme, there will be less scarring in the wound. They are used clinically in dermal fillers, viscous supplements that are used for the joints, and vitreous substitutes. They form hydrated gels due to water retention, and they are key in the glycocalyx structure of the cell. Thank you for watching this video. If you have any query, please write in the comment box.